grad students, right? Yeah. Okay. So this is a little different talk than I normally do, but I know it's always a new audience. So I like to talk to you about my path. I was a grad student here too. So, but I want to tell you about my path, about how I get to my teaching. So the first half is a little about me, and the second half is how do I teach or facilitate learning in the classroom. So how many grad students want to teach maybe down the road? Okay. So hopefully from this talk is that you might be able to get some ideas of how to use active learning in the classroom, or you may pick up on ideas that your faculty are already using and you don't know are active learning. You might learn that from this talk. So I want to tell you about how I came to having a 100% active learning course, and it comes from my background in the arts. So, and I'm going to tell you a little bit about what I do in my lab too. I know that a lot of people are in labs in here too, and to show you how I arose at that. So my lab works on cell division, but I came from a family of artists. So here's sort of my outline. I'm going to talk about my genetics, because I'm a geneticist, talk about my science, how that went into science as art, and then into the teaching. So here's my genetics. I grew up in a family of artists. If anyone knows me, I budded right from my father directly, a crazy personality. So my father was a sculptor and just about every artist, but he also worked as a medical illustrator. So then I knew as very young that you could do science and art, and that was actually important. Most people know that too. But along the ways of my training, I lost that sense of art. He also was an anatomy educator. He taught medical students how to draw bones and stuff like that. So very unusual upbringing and unusual parent, but I didn't know the difference between art and science until I left and went to school and went to college. My mother was a ceramist and art educator, and all my brothers and sisters are graphic designer, industrial designers. So I look like the black sheep here, and someone who's different. I'm unusual, right?
You're also color.
I'm also colored, right? So I ended up the scientist, right? So how did I end up the scientist? I am an artist genetically, but how did I become a scientist? So this is a little bit about this was actually in my home, in my basement. We had an art studio. There's my father, one of my favorite pictures of my father. He was sculpting the bust of his football coach. So my dad he played in the Orange Bowl. My dad was a very winning player center for Jim Brown. So yeah, unusual background. So an artist who's a football player sculpting his coach. He's one of the winningest coach at Syracuse, and this is actually bust is in the Syracuse Carrier Dome where I went to school. So almost our entire family went to Syracuse because my father played football there, who was also an artist. A lot of people at that time in the late 50s and early 60s were actually artists who were on the football team, which is sort of switched. And so we had this unusually unsterile home, right? We had a lot of clay, a lot of plaster in my home. I also was surrounded by a lot of interesting people from all over the world that studied with my father. So here I am at 2 years old. This is in the 70s, mind you, lots of hippies, right? People who are interested, but there were musicians, there were sculptors, painters, philosophers, Zen masters. A lot of people came to study from my father over the world. So it was like being in college, but at home. It was really neat. I got to meet people from all over the world. And so this was my life. This was normalcy for me. You know, it was just very, very, for me, normal. But when I left, I realized how unusual it was and how unique that it was and how beneficial it was for my life. In the back of this is just an example of things that are on, I live in a normal house nowadays. I don't have sculptures everywhere. We had sculptures in our backyard. So this is a view to the backyard, stuff that my dad would always learn how to do something. He would pour concrete. I learned how to do all of that stuff. So having this around my house was very unique, and it was like living in a museum. And that's just a taste of my life. So how the heck did I end up a scientist with this unusual environment that I lived in? So now that you know a lot about me, a little bit, but you know that I have this unusual upbringing, what kind of science would I possibly do? So when I was an undergrad, I saw a picture like this. So this is a one-cell C. elegans embryo in mitosis with chromosomes labeled. So I fell in love with this. This is a more modern image. I saw it in my textbook. I circled it. I said, this is the best thing I've ever seen. It was like a Picasso. I was struck. I was like, oh my God, it's amazing. And I didn't even know what these things these red things were. These are microtubules. And it took me even to grad school to figure out what these actually do. They're very dynamic dynamic polymers that control the mitotic thing. So guess what? I still work on mitosis, partly because it's so beautiful. And that's, so from an artist perspective, this might be very normal when you see something like this. But if you have that science, analytical background, you want to figure out how things work. And I definitely, that's always with me, but even my father always wanted to figure out how things work as an artist. So that kind of ideas are always tossing around in my head, is that a really, really important part of science is, the beauty. Sometimes you want to figure out how things work, but you don't know how and how to ask those questions. But that's what I do in my lab. So my lab works on a very simple process, most people know. Everyone knows cell division, right? Pretty simple, mitosis. One cell goes through metaphase where the chromosomes light up, and then they segregate them to two opposite poles of the cell, and that's important for growth and development. And then when this process goes wrong, you can end up with a tumor. Everyone knows you can get a tumor and a cancer, that's pretty obvious. Birth defects like trisomy 21, where extra chromosomes go, that's another cell division defect. Other people probably also know that stem cells arise from an asymmetric division. So that's components on one side of the cell or the other, and that's really important of how you get stem cells. Probably what you don't know, and things that I'm working on in my lab, is that in neurodegenerative disease, like Alzheimer's, is that the glial cells are multinucleate. And this is only one case of it, but it's something I'm working on in my lab. And so what that struck me as interesting is because those two nuclei that are bagged up in the glial cell suggest that there's a cell division in the brain. Most people think that cell division doesn't occur in the developing brain, but it turns out that in the glial cells they actually do. And it's really important, and I'll talk about that just briefly. So my work was to identify new proteins involved in this process. So I identified 100 of them, and one of them I'm studying in my lab is a gene called ataxin 2. It was isolated from this little pinch point. This little pinch point is called the mid-body. How many people have heard of the mid-body? Some people, right? So not many people know. It's actually considered an organelle after my work, which is pretty cool. But this is actually a very important structure in the final separation of the cell. So if this structure doesn't form, the cells are going to regress and end up with a cell with two nuclei. But we identified actually RNA binding protein that's actually in this little pinch point. And the reason why I wanted to look at it, well, one, no one's been looking at this class of proteins, but when you mutate it, it causes spinal cerebellar ataxia type 2. And that's a mouthful, but basically you've heard of Alzheimer's and Parkinson's. These are ataxias, these belong to family ataxias, the shaking disease. And so what happens in a lot of these patients is that this is a normal brain, but in the SCOT2 affected, it atrophies or dies. And so a lot of people don't know what's happening on the cell biological level, but what happens is basically they have motor coordination, they can't hold themselves up, they have the shaking ataxia. So this is sort of the test. If you ask the child to try to touch a toy or anything, they can't do it because they can't coordinate their neuromuscular function. And so that's what an ataxia is. So, and a lot of people work on ataxias from Alzheimer's, Parkinson's, and to lots of these SCOT2 types of diseases. So the only thing that's sort of known at this point is that in the cerebellum there are these beautiful dendritic arbors, or trees, neurons that look like trees. They're called Purkinje cells. Most people have heard of Purkinje, right? Very famous cells, probably the most beautiful cells in the brain. But in the patients, they actually look like shrubs, right? So they're small and stunted. A lot of people think that there's problems with the neurons themselves, is the reasons why they get small as you age, and that's what causes the phenotype. But less is known about the surrounding environments around that. And so some of the things that were coming up in hypothesis, reason why they're getting smaller, is I'll touch on in the next few slides. So if you knock out the homolog in C. elegans, ataxin 2, we do this in, and it's hard to see, but there's this embryo is right here, so this is the nematode worm I work on. We cut the worms in half and we videotape the embryos that are tagged with fluorescent proteins. So one DNA is in red, plasma membrane is in green. And so we can watch the cell division process. We can't do this in brains, obviously, in human brains, so we can do it in model organisms. The C. elegans is a great system for studying cell division because they're very large embryos and they have stereotypic cell division events. And we can tag things with GFP and watch how things happen. So in a normal cell, you're going to watch the mitotic, so interphase, metaphase, and then the division process. And I love looking at this. I can look at it all the time. That's why you realize you know that you're in a good place, because you've found something you love looking at all over and over again. So the division process is also really rapid. It's only 15 minutes, and so humans it divides for 24 hours, so it's really nice to be able to use a system that's quick. So if you knock out this human disease gene, what happens? There's one defect, because this is actually a meiotic defect, but what's going to happen is their mitosis is going to go through division. The DNA contents are going to segregate, but it's going to try to pinch in, and then it's actually going to fail. So you're going to end up with a bag of nuclei. And so that was a surprise to me, given that no one had ever thought that this was actually regulated in mitosis, something that causes really bad brain neurodegeneration. And so one of the ideas, and long story short, because I'm going to go more into communication here, is long story short is what we think, the hypothesis is that this is a picture of the cerebellum in the brain. It's not mine, but it's a beautiful picture. So these are the Purkinje cells with these nice dendritic arbors. And then these glial cells, so I've been learning more and more about the glial cells. I'm not a neurogeneticist, but I've been learning more and more is that these little red nuclei here are all the glial cells. And they're all surrounding them. This is a better picture of it. So this is basically the neuron. Each of these are newer glial cells. So they're sort of the helper cells in the brain. So this is what we think is actually failing. And so we're collaborating with people who are neurobiologists to actually sort of help us look at this problem. And so when you lack ataxin-2, what we think is that all of this structure is degenerating over time, because these nuclei divide in a certain pattern, and people have studied that for a while. So they divide asymmetrically, and they divide on a certain developmental program. And so that's our hypothesis where we are now. We're using C. elegans to look at this problem and to see what other cytoplasmic defects might be wrong in the ATX2 embryos, and how that could relate to human disease function. There isn't much pathology because there's only so many patients with brain slices, and you can't look at this live, so we can look at that in a live organism. So the other thing about glial cells, which is interesting, is I found in different species, humans have the largest glial to neuron ratio, and it turns out attributing it to creativity and language, is that maybe the numbers of glial cells are telling you that. So I find it really interesting. But there's actually a lot of gliomas or brain cancers that are attributed to cell division genes, but no one's really looked at that. So that's sort of what my lab is working on. But what you can see overall, beauty is what I love, and I love looking at things, and that's how I come to science. And so you might expect that I might make this jump, given what I showed you, is that this looks like art to me, right? And so when I was a grad student here in '97, I started, I think I was a third-year grad student, I started this International C. elegans art show. And so I did it because I come from a family of artists and been curating art shows, and I couldn't figure out why people weren't sharing the beautiful images. In my lab, and probably everyone's had a hard time in graduate school, but when you come from a family of artists and you say, I want to do this art show, people told me I'm making a terrible professional mistake, right? And so I decided at that point I didn't care, right? I didn't give an S, T, right? And now it's an ongoing event, and it got a lot of press, and I was like, wow, it gave me a lot of confidence that I should go on. I didn't, I loved it, but I realized I wanted to share it with, I showed my family, but why can't we share it with others? And so I've been doing it for, since that long, and people have supported it. And it's just been by the efforts of standing up to people and say it's okay to do that, and there are a lot of people who like this stuff. So I'll show you a few examples of what came in, not just me. This is an image from my colleague Abby Dernberg, who's at Berkeley. This is a gonad of the worm, probably the most beautiful gonad you might see. So this is the nuclei labeled in the germline. And so she ended up being my very first faculty member she became a faculty member. She was one of my very first winners of the art show, and it turned out over the years, everyone who entered the art show and won became a faculty member. And so that was a really unusual statistic. But it told me that creativity is really inherent in science, and I've come to know that very well, is that the most creative people also really dabble in arts and music and other sorts of crafts. And so, unbelievably beautiful things come out of your colleagues, and that makes you keep wanting to do these things. Here's something that I did. So some days when you're on the microscope and you're totally you're having a hard day, but you see something that's weird, it's not related to your research, but you snap a photo of it anyway. And so here's my worm that I'm doing. It's on a Petri plate. Does anyone know what these things are? People know Petri plates, right? Well, it could be on a Petri plate by accident. Anything? What do you want to guess what they are? What does it look like to you?
Water.
Water? Could be water. Anyone else?
Yeast.
What? Yeast? Could be yeast, yeah. Anything else? A lot of people think these look like chocolate. Looks like chocolate. It could be coffee beans. I think they look like UFOs, right? So they're actually air bubbles. So the cool thing about that is that you take a picture like that, and sometimes you stand back and look at it. A lot of people see different things in it. But what's interesting is they all look pretty similar, but how can air bubbles do that? So when you make a Petri plate, it goes from a liquid to the solid, and so it had to take the perfect temperature and time and water and condensation to have these beautiful air bubbles have this kind of form. And so those are the things in science that I love, is that you end up having these happy accidents, and then you snap them. And what's great about this, especially for the public, when you're trying to engage the public, most public know what a UFO is, right? And so when I'm trying to talk about what science is, it's really great to hook them, right? And so this is like a great hook. And so this was an accident, but it's a great hook, and this has gone to be framed in one of my art shows. And so partly for the reason is that the public already knows something about the image they're looking at. And so then I can go over here and go, oh, you see this little guy over here? And so be able to tell them is like, this is the model organism I used to study human disease. And so you're able to create a dialogue after you've created that hook. And so I've used art in that way and found out that there's really huge impacts. So anyone been to genetics over there? Anyone seen this? Yeah, so I did this, very proud of it. So from these art shows that I've done nationally, when I came here, this wall was actually all empty. And I grew up in a family of artists. I hate empty walls. And so I walked up to the dean's office and says, I can do something with this space. And at the time, they gave me $15,000 and went down there and did that. And that's like probably the largest amount of money I've gotten without writing a grant. And so I came and I did this and put that up. I think that was in like 2007. And from that, a lot of things happened to me after that. A lot of the people in the art department says, oh my gosh, this is the most amazing thing. You know, they really liked it. And now school buses full of kids come through, and it really is engaging the public about what's going on. And these are images all from the genetics department. And sometimes you just have to, it doesn't hurt to ask. You know, I didn't even ask my chair. I didn't even know there were rules. You know, I just went to talk to the dean and said, I have an idea. So there are, you know, you just have to have enough confidence to go and do it. And I've hit a lot of walls, like I said. A lot of people don't appreciate it too, but sometimes people are out there really like a good idea. So from that led to the tiny scientific art show, which ended up being in the Dane County Airport. These are images from all across campus that were framed. The chancellor at that time actually also paid to frame these and put this, and also the Alumni Association thought as a benefit because it's the science that's going on campus. And that we can market science through the arts. And it ended up getting lots of press with McNeil Lair is the show I hated as a child. But when you get press, it's always nice, good press. And NPR, and it ended up touring. It's been a National Science Foundation, Milwaukee Art Museum, and a lot of different places. So that sort of exploded a lot of opportunities for public engagement that I really liked, and then even more interactions with other artists. And so, has anyone heard of the Cool Science Image Contest? Yeah, so I'm in the part of helping start that too, and I'm a judge, so please enter. This won last year. I was amazed. This is actually blood vessels in the eye. I was overwhelmed. I thought it looked like a Monet's water lily. Most people think that. I just And that's something they didn't use in a journal article, but oh my gosh, to have that taxpayer money go to waste, that we could see this kind of stuff. I think it's just, and you know, her PI didn't even know that she submitted it and ended up winning, and she didn't even think, had the confidence of win. But beautiful images come out of things you least expect. So yeah, if you're doing the kind of science, you know, submit. It gets you a lot more press than you think. The other thing I do is I have arts students in my lab. I started with Chanel. Chanel's a performance artist. She was my first artist in the lab. She came to me and heard about me that my lab was open to artists. I would describe her as the Lady Gaga kind of character, but even better. She's very interested in toggling between science and art. She came to my lab and looked at the C. elegans, the skin, the cuticle, and this is what it looks like, and she was inspired. She made fabric from that inspiration, right? So a lot of new ideas come from the interface of stuff, and she was the very first undergrad to actually perform at the poster session on campus, and she did her performance, and it was all about, and she actually won a Guggenheim Fellowship, so for artists that's a really big deal, and so it was called Differentiate. So she got quite a lot of funding for it and quite a lot of rapport, and got to go to New York City and do her performance and all that kind of stuff. So that's been really cool interaction. And my students, although some students were like, this is crazy, they end up really learning a lot from having the artists in the lab, because their path is totally different than the one of a science student often. And so some students, you know, Chanel, for example, is really good at Illustrator. My students weren't. Or they're not good at seeing some things. Or they look at something and it's not what, you know, they don't have any biases for what's in the lab, you know. And so they take more chances, and it's nice. So there's a beneficial experience for both my lab and the student. I've taken, my first grad student I've had in the lab was Angela. She's a photographer, so if you've been on the second, has anyone been on the second floor of genetics? I don't know. This is on the second floor, it's near my lab, you can come up at any time. She was a photographer by trade and had a lot of experience in museums. So she came and took pictures of organisms and patterns that she saw in my lab, the zebrafish lab, mouse lab, and fly lab, all on my floor, and put together you know, what she saw in the manner that she wanted to do. And she also has a background in old photography and antiquing, and this is also on three-dimensional, so it's on wood, so it sticks out at different things. But it's also a play on a scientific journal article montage, right? So there's the science and art all intertwined there. It's one of my, I really enjoy it because it's not how scientists would take photos, it's more of an artistic flair. But it's great to engage the public about what a zebrafish is by making it more friendly through the arts. Her opening, her MFA or MA opening on the floor brought in 150 people from the public that never even been into a science laboratory before. So that was huge for me to see that. It was huge for the people to think that they couldn't enter a science building or even come into a laboratory, that people were mortified. And so this is where the power of art creates that friendly environment, so that, you know, people can say, you know, what is this, and what, you know, where is my taxpayer money going, and what are you doing, and what impacts are you making, and interfacing with the public more through simple, you know, art communication. So what have I learned so far from all these experiences? Well, like I said many times, you know, science art really can inspire the public really quickly. You know, anyone who sees that gets overwhelmed and starts to ask questions. And the other thing is, art is able to communicate complex ideas to a wide variety of audiences very simply. It doesn't matter who you are. Most people have been in an art museum. Not many people have been in a science museum, but art is a great way to do that. And then working with the artists in the laboratory inspires new ideas on both sides, not only to the artists, but to the scientists. And I really think that engaging the public should be an integral part of everyone's research program, because there's benefits to the public. And as given the current climate, you can see we need more of it, obviously, more people doing it. And there are easy ways as simple as science art can actually do that.